Welcome to PW English YouTube channel. I'm Shalini Somashekhar, your botany teacher. I welcome you to this lecture in the UMEED NEET English batch. Today we're going to discuss the chapter Principles of Inheritance and Variation. This is the first chapter in the second unit of your 12th grade syllabus, which is Genetics and Evolution. Under this unit, you will have three chapters: Principles of Inheritance and Variation, Molecular Basis of Inheritance and Evolution. So yeah, this is the first chapter. It carries high weightage. As you can see, the number of questions that have been asked from this chapter over the last few years, roughly around 5 to 6 questions have consistently been asked. And another thing is, though we teach it under botany syllabus at PW, questions from this chapter can be asked both under botany as well as zoology sections in the actual NEET exam. So yeah, let's get started. This is the syllabus, the latest NEET syllabus for 2025 that has been released by NTA, and there is no change in the previous year's syllabus and this year's syllabus. And if you have a question whether to refer the old NCERT or new NCERT for this chapter, you could refer either. After rationalization, nothing has been deleted from this chapter, so either of them will work. Okay? Yeah. So what is this chapter all about? As the name of the chapter itself indicates, we are going to learn about inheritance and variation and what are the principles that govern it. And this is a chapter under genetics, the first chapter under genetics. So let's just understand what genetics means. I'm sure you've heard about this term in your lower grades as well. Genetics and heredity are things that are commonly used in everyday language, right? So genetics, similar to your morphology, anatomy, physiology, and histology, is a branch of biology. And this branch of biology deals with inheritance and variation. Two very important aspects: inheritance and inheritance and variation. See, today we know that a dog will always give birth to a dog and not a cat because it transfers its genes to the offspring. But back very early on, humans did not have much idea about what genes are, what chromosomes are, how sexual reproduction occurs. And people always pondered upon, why is that an elephant always gives birth to an elephant calf and not like a rhino? How exactly does that information transfer happen? How are the characters of offspring kind of similar to the parents, right? And also, if you think about it in terms of plants, when you sow a mango seed, you always end up getting a mango tree and not a banyan tree, right? What is present within the seed, or how exactly is the parent plant ensuring that its baby or its seed also is going to look very similar to it and not like some other plant? So people started thinking about that, and that is how the uh, interest towards how characters get passed on from one generation to another arose among people. So if you think about it, like I already mentioned, in genetics we're going to study about two concepts. One is inheritance and the other one is variation. Now, what is inheritance? Let's understand what inheritance means. Inheritance is a general English term. You may have heard people say that I inherited some land from my father, I inherited some property from my uncle, I inherited this jewelry from my mother. So when they say inherited, they are basically saying that something that was not theirs, it was part of a previous generation, it has gotten passed on to them, right? That is what inheritance means. If you apply this to the genetics context, we are talking about how inheritance of characters happen. If you notice, you may have had people tell you that you look a little like your mother, or you look like your dad, or you look like your aunt. So what do they mean when they say, let's say you look like your mother? That means some of the characters that your mother has, you are having. Probably she has straight black hair and you have straight black hair. That is one character. Probably her eyes color could be dark brown and you have dark brown eyes. So that is one character. We say that with respect to genetics, whenever characters like these are passed on from one generation to the next, the process by which that character get passed on, that is called inheritance. Okay, so there is a process that happens when my characters will get passed on to my child. The process by which my characters are passed on to my next generation, That process is called inheritance. My child inherits my characters. The process by which he or she inherits my characters is what we call inheritance. Now, what is variation? Variation again is a very general English term. Variation means variety or difference, you can say. Even though my child, I give birth to my child will not be 100% like me or like the child's father. Like you could have some characters of your mother, you could have some characters of your father. You're not 100% like either of them. You might look very similar to one of your parents and not so similar to the other, but you're not 100% like either of them. Even though the child is a combination of characters of both the parents, you're not 100% like either of them. If you think about a family that has more than one child, two siblings are there, they're not identical twins, let's assume. When they're not identical twins, though they are born to the same set of parents, still there will be difference among the two children, right? They're not 100% alike. So even though characters are getting passed on, the offspring will not be identical to the parents. There will be some degree of variation. To what degree there is difference, that is what we call as variation. So in this chapter, we learn about what are the principles that govern inheritance and variation. Principles means what? Principles is rules that govern something, right? Like you have like laws of gravity and laws of motion and those kind of things that govern how those processes work. Similarly, how does inheritance work? What are the rules that govern inheritance, passing on of characters? What are the rules that govern variation? How exactly is that variation generated? Why are they not identical? So what exactly happens? All of this, all of the rules and principles that govern inheritance and variation, we'll understand in this chapter. Okay, so we have to first know the definition of inheritance and variation. I've already mentioned inheritance is a process by which what is getting passed on from one generation to another? Characters are getting passed on. What do we mean by characters? How tall a person can grow, what will be the color of the skin, what will be the color of your eyes, what will be the texture of your hair. These are your characters, right? So how from one generation to the next? You can say one generation to the next, parents to the offspring, parents to the progeny, anything. All of that is the same, right? So by what process the characters are getting passed on from parent to progeny is called inheritance, and that is the basis of heredity. You may have heard people, laymen also use the term heredity, right? If someone's like too tall, they say, oh, that's heredity, that runs in their family. Everybody in their family is very tall. If somebody has like thick, dark, curly hair, then they say, oh, it's heredity, that runs in their family. That just means it's getting passed on from one generation to another. So inheritance itself is the basis of heredity, right? Now, what is variation? Like I already mentioned, two parents, they give birth to children. None of the children look identical to either of the parents. There is no similarity between siblings also if they are not identical twins. Okay, so that degree by which the children differ from their parents, that is what we refer to as variation. All right. Now, we know that Mendel kind of scientifically laid the foundation stone for the field of genetics. It's not that people before him weren't aware of how this works. Humans as early as 8000 to 1000 BC knew that in populations there will be variation. You know how much 8000 BC is? That is before the start of what we count right now, 2024-25. This is like 2025 years, like 8,000 years before that. That's like 10,000 years from now. Humans, as you know, are inquisitive beings. That is around the time probably when they started rearing animals and agriculture and all of that. So they knew that in a population, variation is always going to exist. Like for example, when you take an example of people rearing cows, and let's say in a household there are 10 cows and there are like 4 or 5 bulls, they knew that within that population of cows there is variation. Not every cow is going to yield the same amount of milk. Probably one will give 5 liters in a day, one might give 12 liters in a day. So among that population itself variation exists, they knew it. And they also knew that the reason for this variation is sexual reproduction. They had no clue about what happens during sexual reproduction, what happens during gametogenesis, but from their observation, they were aware that variation exists, and the reason for that variation is sexual reproduction. And of course, humans being humans, we know how to exploit things for our own good. They exploited that knowledge and they exploited the cattle that they had by selectively breeding them. What do I mean by selectively breeding them? I will select whichever among the cow is the highest milk yielding cow. I will select that, and I will select a bull which is hardy, kind of work in the field. I will select that, and I will have them mate. And of their offspring, whichever offspring has my desired combination of characters, I'm further going to select them. So this is what they did. And that is how we obtained many breeds of cattle in India, such as the Sahiwal cows in Punjab. Okay. So humans knew as early as 8000 to 1000 BC that one of the cause of variation was hidden in sexual reproduction. They exploited the variations that were naturally present in the wild populations of plants and animals to selectively breed and select for organisms that possess desirable characters. For example, through artificial selection and domestication from ancestral wild cows, we have well-known Indian breeds today, such as the Sahiwal breed that we have in Punjab. Okay? Yeah. So about who Mendel is and what he did, you may have learnt in your lower grades. Mendel is called the father of genetics, and he proposed some laws of inheritance. Okay, so what did Mendel do? He was studying inheritance. He was very curious about how characters get passed on. So he spent 7 years of his life studying inheritance in pea plants. The scientific name of pea plant is Pisum sativum. So he worked on the garden pea plant, Pisum sativum, for 7 years from the year 1856 to 1863. Okay, you must remember this. It was asked one year in NEET, which year he worked. How exactly he performed his experiments, what were the results of his experiments, we'll discuss that later. First, I'm giving you like an introduction to the father of genetics, Gregor Mendel. Okay, so what he did was he performed experiments using a large sampling size. You know, when the sampling size is large, your data becomes more credible. Sampling size means how many plants he performed that experiment with. That is his sampling size. It is said in some books that he used more than 1000 plants to get his results. Because he got the same results every single time in all of them, his data was more credible, right? When you consistently prove yourself, you become more credible. Just because you scored like 100% marks in mathematics in second grade, you can't call yourself a math topper today, despite scoring 35 out of 100, right? So if you've consistently been scoring similar kind of marks over the years, you can call yourself like a genius at math. That gives you more credibility. Just because you did something once, you don't, I mean, it's not that credible. I hope you're getting my point. So since he had a large sampling size and he consistently got similar kind of results in all of them, his data is more credible. And also, because this is inheritance experiments, you can't perform like one cross, perform one experiment with two parents and get one result and say, okay, see, this is how it worked this one time. This is probably how it will work every single time in every single organism. Let's call this a law. It is not credible in that case. After one cross, you got one particular result. You will test the same thing in the next generation and the next generation. If you're consistently getting similar observations, similar results, again, that will give you more credibility. So what set him apart is that he confirmed whatever inference he got from one generation on successive generations as well. So again, that gives more credibility to his data. Another unique thing about Gregor Mendel is he was good at math, physics and biology, all of them. So he kind of tried to explain biological phenomena, biological concepts by integrating them with mathematical concepts like statistical analysis and mathematical logic. So it's very rare that we see an intersection happening between mathematics and biology. But between bio and chemistry, we talk about biochemicals and the structure of our molecules. Between math and physics, there is integration, but between math and bio, it is rare. Unless we're talking about some graphy plotting something, we don't really get math into biology that much, right? So here, this was kind of unique because he applied mathematics to solve the problems in biological phenomena. Okay, and yeah, these are some contributions of Mendel. So like I mentioned, he chose pea plant for his experiment. Let's understand what could be the reason, what are the advantages of selecting pea plant as his experimental organism. Why did he not select a mango tree? Why did he not select a banyan tree? Why did he not select banana? So there are so many options. Why was pea good for his experiments? Why was that a good choice? Firstly, the pea plant's lifespan is very less. Okay, it is an annual plant. It will complete its life cycle within 1 year itself. So that is a good thing, right? If you're studying about inheritance, you want to see how characters are getting passed on from one generation to the next. If it takes that plant 6 years to produce flowers and fruits and seeds, what are you going to do for that 6 years? You want quick results. You want the next generation very fast so that you can do your studies. Over here, since the lifespan is less, you will get the next generation very quickly so that you can perform your experiments and make necessary observations and inference from the experiments, right? Short lifespan always helps in genetics study, right? So that is one advantage. It has a short lifespan. Next, you don't need like a lot of labor and maintenance to grow this plant. You know, pea plants kind of are like creeper climber kind. You don't need a lot of labor, and in a small place itself you can grow very many plants. Okay, so many plants can be grown in a small area, and within one plant, within one species, you can see a lot of contrasting characters. Like when it comes to the color of flower, it is violet or it's white, which is very contrasting. It's not like light orange, dark orange, where there is not much contrast. Violet and white are very easily distinguishable. The color of the seed, either it's yellow or it's green, very easily distinguishable between yellow and green. It's not like light green and dark green. That is a difference, but it's not very contrasting. It's not like a stark contrast there. But there are many characters in the pea plant which are very highly contrasting, which is useful for our studies. Okay, so it had a lot of variety of characters that were contrasting. Next, in each plant, many seeds are produced. Many flowers will be there, many pods will form, many seeds will form. If there are many seeds, again, like I mentioned, the credibility is more, right? You're getting many children from one plant. Many seeds are like babies for plants. So you're getting many of them. You can analyze the characters better. That is another advantage. And finally, if you allow these pea flowers as they are, don't disturb them at all, it will undergo self-pollination. But it's also very easy for us to cross-pollinate pea plants. When you're performing genetic experiments, of course, you will have to cross them. And sometimes you will have to allow them to undergo self-pollination. So both of them are easy here, not much of a hassle. Because of these characters, Mendel chose pea plant, okay, as his experimental organism. So what did Mendel study here? There are many characters in a pea plant: the root system, the stem, the shape of the stem, the leaf, the position of the flower, the color of the seed, the color of the flower. There are so many characters on a pea plant. But Mendel chose 7 characters in a pea plant, and each of these characters, existed in 2 contrasting forms. We call them traits. Like for example, flower color is one character. Flower color existed in 2 varieties, 2 forms. One, either it was violet or it was white. So these are called traits. So he chose 7 characters. Each of these 7 characters had 2 opposing traits or contrasting traits. So he chose a total of 7 characters and 14 traits. Alright. Character is a heritable feature that varies among individuals, and trait is a variant of a character, like I already told you. Right. Yeah. So Mendel conducted his experiments using true breeding pea lines. What is true breeding pea line? True breeding means you're allowing them to undergo self-pollination generation after generation, such that if I'm taking like a plant that produces violet color flowers, purple color flowers, I allow it to undergo self-pollination for several generations. up until the point I'm sure 100% that if this plant undergoes self-pollination, I'm only going to get violet colored flowers. Up until that point, if you are self-pollinating it, then what you obtain is called a true breeding variety or a pure line. Alright. So what exactly is a true breeding line? When I explain to you homozygous, heterozygous conditions, you'll understand. For now, just know it is true for its trait. If it is producing violet, if a plant is producing violet color flowers, if you allow it to undergo self-pollination, then you can be rest assured that all its offspring are going to produce violet color flowers. It is going to stay true to that trait. That is called a true breeding variety. What exactly, how exactly it ensures that only violet color flowers are produced, we'll understand later. For now, just remember that is what true breeding variety means. If a dwarf plant is there, like a short pea plant, you allow it to undergo self-pollination for several generations, then at one point you will be sure that if this plant is allowed to undergo self-pollination, then for sure it will produce all dwarf plants. It is true to that trait of being dwarf. Okay. So true breeding line is one that, having undergone continuous self-pollination, shows the stable trait, inheritance and expression for several generations. So Mendel, I told you, he chose 7 characters and 14 traits. For each of those 14 traits, he selected pure breeding varieties that were true to that trait. Okay. Now, which are the 7 characters that Mendel selected? One for stem, okay, the height of the stem. 2 characters for flower. One is color of the flower, one is position of the flower. 2 characters for fruit or pod. One is color of the pod, one is shape of the pod. And 2 characters for seed. One is color of the seed, shape of the seed. So stem height, flower color, flower position, pod color, pod shape, seed color, seed shape. These are the 7 characters, and each character had 2 contrasting traits. No in between, no intermediate, 2 stark contrasts. The stem height, either it could be tall or it would be dwarf. There is no intermediate height. Flower color, either it would be violet or it would be white. Flower position, either it would be axial or it would be terminal. Pod color, either green or yellow. Pod shape, inflated, which means it is full, like a balloon with air filled inside. Constricted means there are narrow regions like this. Here you can see it becomes narrow, right? That is called constriction. So it can be constricted like this. Color of the seed, either it can be yellow or it can be green. Shape of the seed, either it's round or it has like a wrinkled appearance. So these are the 7 characters and the 14 traits that were selected by Mendel. So what exactly did he do here? Again, this is another slide showing you the same thing. The characters and the contrasting traits are listed here. For your reference, you'll get it in your notes. Yeah. So what exactly did Mendel do? In all of Mendel's experiments, he did the same steps. What is the first thing? He will select 2 parents. Okay. Both of these parents will have contrasting traits. In the first step, selection of the parents is important. That is the first step. After he selects these parents that have 2 contrasting traits, both of them have to be true breeding varieties or true breeding lines, he crosses them. Okay, that is the second thing. First is selection, second he crosses them. How does he cross them? He performs artificial hybridization. How is that done? We've learnt about it in the previous chapter. Pea plant have bisexual flowers, which means they have stamens as well as the carpel as well. So if cross-pollination, artificial hybridization has to happen, you have to designate one plant as male and one plant as female, right? Whichever plant you've designated as female, in this case it is the plant that is producing violet color flowers. Male is the plant that is producing white color flowers. Okay. So if this is female, you have to remove the male part from it. If I'm designating this as the female, I will remove the male part from it. Which is the male part? It is the stamen. How will I remove it? By cutting it off before it matures, before the anther dehisces. So I will cut it, and that process is called emasculation. After I'm cutting it, I have to ensure that pollen from this only falls on this, not some random pollen, not the same pollen. So after I emasculate it, I will transfer pollen grains from this to this, and to ensure that no other pollen will come and fall on it, I will cover it with a bag, and that process is called bagging. and you will allow that flower to develop into a fruit, and then you will get the seeds, and that is your babies from this plant. That is the next generation. So he gets the next generation. The next generation is called the first filial generation. In short, we call it the F1 generation. So this is the pod that you got. These seeds are the babies. You sow the seeds, whatever plants arise from those seeds, that is your F1 generation of plants. Alright, so far what have we done? We've taken two plants that are pure breeding with contrasting traits. We've crossed them by artificial hybridization. You've obtained seeds. That is your F1 generation. You sow them, you plant them, you get plants, and that is your F1 progeny or your F1 generation of plants. Now you will allow these F1 generation plants, or your F1 hybrids you can call them, to you allow them to undergo self-pollination. At first you cross-pollinate them, then you allow them to undergo self-pollination. Okay, so that is basically what Mendel did. See here, this will be allowed to undergo self-pollination in the next step. Okay, now Mendel did this for one character at a time, which we call monohybrid cross, and he also performed these experiments, similar experiments, same steps, considering two characters at a time, that we call dihybrid cross. First, let's understand monohybrid cross. The character that I am taking now is the height of the plant, the stem's height. It can either be tall or it can be dwarf, right? These are the two contrasting traits. So I've selected two parent pea plants. One is tall, one is dwarf. This is a pure breeding tall plant. This is a pure breeding dwarf plant. So I've selected. The first step is done. What is the second step? The second step is to allow them to cross-pollinate them by artificial hybridization. When he crossed a pure breeding tall plant with a pure breeding dwarf plant, all the plants that he obtained in the F1 generation were tall. Okay, what did I tell you are the steps? Selection, cross, F1, allow F1 to undergo self-pollination. So all tall plants he got. In the F1 generation, whatever plants he got, he allowed them to undergo self-pollination. When they underwent self-pollination, whatever offspring you will get from that is called your F2 generation. And in the F2 generation, he noticed that there were tall plants and dwarf plants, both of them. And he noticed that the proportion of tall plants and dwarf plants was 75%, 25%, which is, the ratio is 3:1. Okay, 75% of the plants were tall and 25% of the plants were dwarf. Okay, so this was his observation. All right. So what did he observe? He observed, the first observation is that in the F1 generation, only one of the parental traits was expressed. See, the parent was tall, there was one tall parent and there was one dwarf parent. In the F1, all of them showed only this parent's feature, character, right? Only this trait was expressed. That is his first observation. What is the second observation? In the F2 generation, both the traits were expressed. In the F2 generation, we saw how both the traits were expressed, and they were expressed in 3:1 ratio. What is the third observation? It did not show any blending. Blending means what? Showing intermediate characters. If there is a tall plant and a dwarf plant getting crossed, if there is intermediate height plant, then you can say that there is a intermediate trait. But here, the plant was always either tall or dwarf. You don't have like an intermediate height, right? So they always showed the same trait as that was there in the parents without any blending. This was his observation from his experiments, right? Now, what did he infer from his experiments? Okay, he performed the experiments, he made these three observations. What did he understand and what did he infer from those results? Firstly, Mendel said that there is something in the parent plant which is getting passed on as it is unchanged to the next generation. There is something, we don't know what that is, we don't know if it's DNA, it is chromosome, what it is, he does not know. He said that there was something that was being stably passed down, unchanged, as it was in the parent, exactly as it was, it is getting passed on to the next generation. And how is it getting passed on? It is getting passed on through the gametes. And he called this something that is getting passed on, he gave it a name, he called it factors. Today we call them as genes, but back then Mendel gave it the name factors. Okay? And today we know that genes are the units of inheritance. Now, genes will have different variants. For example, hair color is one character. The color of your hair is controlled by a gene. Now, hair color can exist in different varieties, right? Hair color is a character. Hair color is a character. Now, this hair color could be black like it is in most of us. It could be brown. People could be blonde. Or there could be redheads. So you have different varieties in hair color. These are the traits. Okay. So height is the character in pea plant, whether it is tall or it is dwarf. These are the traits. In the characters that Mendel chose, he selected characters that had only two contrasting traits. But it's not necessary that every character in every organism will have only two contrasting traits. Okay? Like if you see in hair color, we have 1, 2, 3, 4. These are the 4 different contrasting traits that we have for a character. Now, this character is controlled by a gene. Gene is nothing but DNA. Right? Today we know that it is nothing but DNA. And in this DNA, there is a sequence of ATGC base pairs. You've learnt about it in the chapter biomolecules. Now the gene is always responsible for hair color. But this gene's sequence can vary. If it is this sequence, then probably it will express black color. If in this sequence, instead of C here at this position, if there was a T, probably it will express brown color. If over here instead of AT, if there was a GC, probably it will express blonde color. I'm not saying that is the exact sequence. Genes will be very long. I'm just giving you like a reference. Genes control a character. Genes can exist in different forms. How are they different from each other? Based on their sequence. Different sequences can give rise to different phenotypic traits. Okay. So the gene is the same. It can be present in different forms. Each form will give rise to a different trait. Now, the different forms of this same gene, form 1, form 2, form 3, form 4. What are its 4 different forms? Different forms of the same genes are called alleles. One character will exist in different varieties, that is called the trait. One gene can exist in different forms with slight changes and modifications in the DNA sequence. They are called alleles. All of these alleles are going to express the same character, hair color. Because of the difference in their DNA sequence, they could either be black, brown, blonde, red hair, whatever. Since we are diploid organisms, P is also diploid organisms, which means we have 2 sets of chromosomes. We receive one set from the father. We receive one set from our mother. So we have 2 alleles for each gene. If your father had Let's say case A, father has black hair, mother has brown hair. It's possible that the child will have one copy of black allele, one copy of brown allele for hair color. Let's say in another condition, like most of us, father also has black hair, mother also has black hair, then probably the child will have two copies of black allele for black color hair. Though in one population of humans, all of these traits can exist, black, brown, red, blonde, all of these can exist. But one individual will always have two alleles only for each trait. Among that, one is obtained from the father, one is obtained from the mother. It is possible for both the alleles to be dissimilar, they are not the same. It is possible for both the alleles to be similar also. Whenever there are two alleles that are not the same, dissimilar, we call it heterozygous condition. Whenever they are similar, we call it homozygous condition. Hetero means different. Homo means same. During the formation of the zygote, two different alleles came together and gave rise to the heterozygote. During the formation of alleles, both the alleles from the parents were similar. It gave rise to a homozygote. This is how it is. Okay, so I have two alleles for every character, one that I got from my mother, one that I got from my dad. Okay, so that's how it is in all diploid organisms, not only humans and pea plants, in every diploid organism that is sexually reproducing. That is how it's going to work. Now, let's understand this better with the help of using some letters and symbols. Okay, so we will consider the same character again, which is the height of the plant. I have a tall plant and a dwarf plant. When I mentioned that this tall plant and this dwarf plant, both of these are true breeding lines, what I meant is that they are both homozygous. Right now, I'm going to use letters to represent alleles. This height of the plant character is controlled by one gene. Now, this gene has two forms. These two are two different forms of the same gene. So, these are the alleles of this gene. Capital T is used to represent tallness, lowercase t is used to represent dwarfness. So I know this is tall and it is homozygous. So this should have two alleles that are capital T. This is dwarf, so it will have two alleles that are lowercase t. Okay. So now we will perform the same experiment that Mendel did using these letters. Okay. Another thing that I want to tell you is, okay, so let's say there is capital T and there is lowercase t. These are the two alleles. T is responsible for tallness and lowercase t is responsible for dwarfness. Right? Now, maybe when we do this, you'll understand. Okay, let's go ahead and do this and then I'll come back to this. Yeah, let's start off with the parental generation. Parental generation. Okay. We'll write down the phenotype and the genotype. Phenotype is what you can see. Like physically, you can see that the plant is tall or dwarf, right? That is phenotype. What are the alleles or genes present inside it? Something that you cannot see, that is its genotype. Okay. So in the parental generation, firstly, what did we take? We took a tall plant and crossed it with a dwarf plant. What was the genotype of the tall plant? Capital T, capital T. The genotype of the dwarf plant was lowercase t, lowercase t. Now, when this performs, when we are artificially hybridizing it, within the pollen grains and within the ovule, gametes will be formed, gametes will be haploid, so only one of these alleles will be present in the gametes. When these two gametes come together, we will get the F1 generation. In the F1 generation, the genotype would be capital T, lowercase t, this capital T and this lowercase t. And its phenotype would be tall. Remember, I told you that in the F1 generation, all the plants that we obtained were tall. So in this F1 generation tall plant, this would be the genotype. This is important, remember it. Okay. Now, what did they do next? They selfed the F1 generation. On selfing F1 hybrid, what happened? The phenotype, it's a tall plant, you're selfing it. So both the male and the female will obviously be tall. What is its genotype now? It's not capital T, but it's capital T lowercase t. It is heterozygous, capital T lowercase t crossed with capital T lowercase t. Now that it has two different types of alleles, it can form two different types of gametes. Okay, so capital T, lowercase t, this also can form capital T, lowercase t. So what are the possibilities of genotype, different phenotypes? To understand that, we construct what is called a Punnett square. Punnett is the name of a person who came up with this idea of analyzing different crosses, finding out what are the different phenotypes and genotypes that are possible. His name is Reginald Punnett. He was a British geneticist. Okay, so this is tall male gametes I'm putting on one side, female gametes I'm putting on the other side. So you know, if these two come together, it will be capital T, capital T. This and this comes together, capital T, lowercase t, capital T, lowercase t, lowercase t, lowercase t. Right? So here you have a tall plant, a tall plant, a tall plant, and a dwarf plant. If you look at the phenotype, there are only two different types of phenotype. You have 3 tall is to 1 dwarf. It's important for you to remember this. If you look at the genotype, there's 3 different types of genotype. You have 1 capital T, 2 capital T lowercase t, 1 lowercase t lowercase t. So the genotypic ratio is 1:2:1 and the phenotypic ratio is 3:1. Alright, now the same thing I've represented here. It's all typed for your reference. You'll get that in your notes. Now based on these results, he proposed some principles that we today call laws of Mendel, laws of inheritance, Mendel's laws of inheritance. He proposed 2 laws based on this monohybrid cross results. The first law is called the law of dominance, or you can simply call it the first law of Mendel. Okay, so it's called the law of dominance. What this states is something that we've already discussed. Characters are controlled by discrete, unchanged, stable units that he called factors. In diploid organisms, these factors are always going to occur in pairs. One we get from the mother, one we get from the father. Whenever there is a dissimilar pair of factors, like there was here, this is dissimilar. Or you can say whenever there is heterozygous condition, There is information for tallness, there is information for dwarfness, but only tallness character will express and the plant will be tall. The plant cannot be simultaneously tall and dwarf at the same time. Only one of them will get expressed. Whichever one gets expressed is called the dominant trait. Whichever one will not express in the F1 generation, that is called the recessive one. So whenever there is a dissimilar pair of factors, one member of the pair dominates, that is called the dominant trait, and the other one that does not express is called the recessive trait. So in the experiment that we saw previously, the tall trait was dominant and the dwarf trait was recessive. The tall trait, dominant trait, always can exist as homozygous condition, in which case it will be capital T, or it can be in heterozygous condition, capital T, lowercase t. But dwarf is always homozygous. Lowercase t. If we are performing cross for any character, how will we choose the letters in Mendelian crosses and the experiments? Here you can see that this is the dominant trait. Dominant trait is tall. So the first letter is T. You will choose this letter, T. If T is capital, that represents the dominant allele. For dwarf, we don't use D. The same T, if you make it lowercase, that will be recessive. Okay, so this tall trait is represented by, the trait is represented by capital T, dwarf is represented by lowercase t. That is how it works for every other character as well. So when he performed similar experiments for all the other characters, he found out which is the dominant trait, which is the recessive trait for all the 7 characters. Of course, we know what it is for the stem height. Whatever is on top is the dominant trait, whatever is at the bottom row is the recessive trait. For the stem height, tall is dominant, dwarf is recessive. Flower color, violet is dominant, white is recessive. Flower position, axial is dominant, terminal is recessive. Axial means flowers form at the axil of the leaf, terminal means at the tip. Okay. Pod color, green is dominant, yellow is recessive. Pod shape, inflated is dominant, constricted is recessive. Seed color, generally we tend to think that peas are green in color, so probably green is dominant, but that's not the case. In pea color, yellow is dominant, seed color. Yellow is dominant, green is recessive. In seed shape, round is dominant, wrinkled is recessive. Alright, so these are the results that he got for the other characters. You must remember very well. Another law that he proposed, the second law of inheritance, is also called the law of segregation. There is a mistake here. It's not law of dominance, it's law of segregation. Okay. So, even though parents have 2 alleles, like every organism has 2 alleles, during the formation of gametes, both the alleles will not go into the gametes. The alleles will segregate such that only one allele goes into the gamete. We know that, right? We are diploid, gametes are haploid. Only one set of chromosomes, one set of genetic information gets passed on into the gametes. This is what Mendel said. Even though the parents contain 2 alleles, during gamete formation, the factors or alleles of a pair segregate from each other such that a gamete receives only one of the 2 factors. We did see that here. Parents have 2 alleles. Parents have 2 alleles here. But in the gametes, only one allele is getting passed on. In homozygous condition, you get only one type of allele. Whenever it is heterozygous, since there are 2 copies of or 2 different types of alleles, one subset of the gametes will get capital T, one subset of the gametes will get lowercase t. Okay, so 2 different types of gametes can form. Alright, now that's about law of segregation. Now let's understand Mendel's monohybrid test cross. See, I told you how a dominant phenotype can either be homozygous or it can be heterozygous. Okay, for example, let's say one dominant phenotype is tall, height. It can either be capital T or it can be capital T lowercase t. If you look at a tall plant, you can't say, oh, this has two capital T, this has one capital T, one lowercase t. You can't. So if you want to find out whether it is homozygous or heterozygous, there is one test you can perform. What you will do in that test is, you will take this plant with the dominant phenotype. It could be capital T or it could be capital T lowercase t. You will cross it with the plant having recessive phenotype, which is dwarf plant. If in the progeny you get all tall plants, that means your plant was homozygous. Here, since it's heterozygous, you have two different types of gametes. Here you have only one type of gamete. If you get tall and dwarf plants in the ratio 1:1, you can say that this plant was heterozygous. By looking at these results, you can say what was the genotype of that dominant phenotype. Dominant phenotype, you can see, if you want to test its genotype, we're performing this cross, so it's called a test cross. Basically, you take the plant or the organism with unknown genotype, but it has a dominant phenotype. You cross it with dwarf plant or the plant that is showing recessive trait. That is called a test cross. Okay. So this was Mendel's monohybrid cross results. Now, similar to what cross Mendel performed in pea plants, many people attempted similar experiments with many different types of plants and other organisms. One such experiment was conducted on a plant called snapdragon. It's also called the dog flower. Scientific name is Antirrhinum majus. Okay, so all of them is the same thing, dog flower or snapdragon or Antirrhinum species. In this plant, the character that they chose was the color of the flower. They took one plant that was true breeding for red flower color, another plant true breeding for white flower color. But in the F1 generation, they expected either this or this. That is what happened in Mendel's experiment, right? They took a tall plant and a dwarf plant, they got a tall plant. They took violet flower plant, white flower plant, they got violet color flower producing F1 generation. Only one of them was expressed. That is according to Mendel's law also, only one of them will express. That one is dominant. But here, in the F1 generation, all they got was pink flowers. It's not like this parent, it's not like this parent either. It's not red, it's not white. It's not like either of them. It's a brand new phenotype. And that phenotype is intermediate. That means the color is in between these two. Right? Pink. If you mix red paint with white paint, what do you get? You get pink. It's like an in-between color of red and white. And when they crossed the F1 hybrid pink colored flower producing plants, in the F2 generation, if they allowed this to undergo self-pollination, not cross, when they were selfed, in the F2 generation, you got the same genotypic and phenotypic ratio, 1:2:1. Okay. So, here one was red, two were pink, one was white. This is the ratio that they got. See, this is the actual plant, dog flower. When red color flower producing plant is crossed with white color flower producing plant, we get a pink color flower producing plant. Let's try to solve this using the same letters that we used earlier, similar letters. Okay, so first we have the parental generation. In the parental generation, what was the phenotype? One parent produced red flowers, the other parent produced white flowers. What could be the genotype? Capital R crossed with lowercase r. Since they are homozygous, they will produce one type of gamete. This will produce capital R, this will produce lowercase r. And what will we have in the F1 generation? The genotype would be capital R, lowercase r, and the phenotype would not be red or white, it would be pink. What are we going to do next? We are going to allow the F1 generation to undergo self-pollination. On selfing F1, what do we have? The phenotype is capital R, lowercase r crossed with capital R, lowercase r. Wait. Pink crossed pink. The genotype would be capital R, lowercase r crossed with capital R, lowercase r. Each one will produce two different types of gametes because they are heterozygous. So you have capital R, lowercase r, capital R, lowercase r. Plot it. Capital R, lowercase r, capital R, lowercase r. So R, capital R, lowercase r, heterozygous, homozygous, recessive. And here we will get, this will be red, these two will be pink, and this one will be white. So you get phenotypic ratio will be 1:2:1. Genotypic ratio also will be 1:2:1. One will be red, two will be pink, one will be white. Capital R, heterozygous, lowercase r, lowercase r. So this is how it is. Since this isn't following Mendel's rule or Mendel's law of dominance, we call this a deviation from Mendel's laws. Okay? So this is called incomplete dominance. Why am I calling it incomplete dominance? Because red is not fully overpowering white color. We are seeing a new phenotype which is intermediate. So it is like partial dominance, not completely dominating. So this is called incomplete dominance. Okay. So I have represented the same thing over here in this slide. Now let's understand the concept of dominance. What exactly is dominance? You know that I already told you that within chromosomes DNA will be present. Within DNA you have the sequence of nucleotides, and for each gene you can have more than one variant which we call alleles, right? So every gene contains information for a particular character or for a particular trait, we can say. Now in some organisms both the alleles will be of the same type for a character which we call homozygous. I've already explained this. And in some people, two different alleles can be present for the same character, two different alleles from the parents, and that is called heterozygous condition, right? So they need not always be identical, we know that. What causes change in the two of them? How are two different? They are different because of their nucleotide sequence. This also I've told you. For example, for example, if you look at two alleles here, one allele has this sequence, this has this sequence, the change is only at this position. Okay, here you have CG, here you have AT, so that is what is causing variation between these two alleles. Now, how did that variation come about? How did it happen? In a population, how are new alleles introduced? That happens because of mutations. So sometimes, let's say in all individuals in a population, initially this was the allele for a particular character. You know how you're constantly exposed to so many different kind of mutagens that can cause mutations. It could be the UV rays from the sun, it could be certain chemicals in your cosmetic products, it could be the smoke in cigarettes. All of these can cause mutations. So when mutations happen, the sequence could change. The change in sequence will ultimately affect the protein that particular gene synthesizes. Every gene controls a character by synthesizing a protein. If the sequence of DNA is changed, the structure of the protein could change. It may or may not change. Okay. Sometimes when there is a very trivial kind of mutation, the protein will not change very much. Sometimes when the mutation is bad, then there will be a change in the structure of the protein. Now let's say one such mutation has happened and here the nucleotide sequence is altered. Okay, so the normal allele produces a normal protein, which in this case is an enzyme. You know what enzymes do? Enzymes are biocatalysts. They will help in the transformation of substrate to product. For them to convert the substrate to product, the substrate has to tightly bind to the enzyme at its active site. And the shape of the protein, the structure of the protein plays a very important role, because if there is any change in shape in the active site, the substrate cannot effectively bind there, right? So in the normal allele, the protein is perfectly formed, the enzyme is perfectly formed, substrate beautifully fits in the active site, gives rise to the products. Now when the enzyme is modified or mutated, there are three possibilities. Firstly, the enzyme could be normal even after modification, because the mutation is not very major, it is a trivial mutation. The enzyme produced could be normal, or there is a small change such that the enzyme produces products, but the products are not produced at the same rate as the normal enzyme. So that is one possibility. So normal or a less efficient enzyme is produced. So this will continue to produce the products. Okay. Here, an allele can be modified such that enzyme is produced, but its shape is so different that the substrate cannot fit in there at all. Products itself are not formed. Or it could even happen such that the allele is modified in such a way that it doesn't produce a protein at all. It doesn't produce the enzyme at all. If the enzyme itself is not produced, then obviously there will be no products, right? So these are different ways in which the allele could be modified. So in major cases, the normal allele will be the dominant allele and the one that is modified will be the recessive one. Now, let's talk about another pattern of inheritance called codominance. This also doesn't follow Mendel's law of dominance, okay? So in Mendel's law of dominance, we saw how whenever there is a dissimilar pair of alleles, only one of them will express, that is called the dominant one. The one that doesn't express is called the recessive one. In incomplete dominance, we saw that there are dissimilar alleles. Neither of these phenotypes will express in the F1 generation, will get a brand new phenotype, and that will be intermediate between two parents, right? Two parental phenotypes. In codominance, what happens is when there is a dissimilar pair of alleles, both of them will simultaneously express. Co means together, right? Both of them simultaneously expressing is called codominance. An example for codominance is seen in the AB blood group of humans, okay? So in humans, we have four phenotypes for blood group. There is A blood group, B blood group. Okay, so it's here. And there's A, B, O and AB. These are the different phenotypes of blood group. What is A blood group? On the A blood group, in people who have A blood group, on their RBCs, they have A type of antigen. If somebody has B blood group, on their RBCs, they have B type of antigen. On people who have AB blood group, they have both A and B types of antigen. In people who are of B blood group type, they don't have A or B. They have no antigens present on them. Antigen is basically a sugar that is present on the surface of RBCs. Okay. So the blood group in humans is a character that is controlled by a gene called I. This I exists in three forms. Capital I superscript A produces the A antigen. Capital I superscript B produces the B antigen. Lowercase I does not produce any sugar, any antigen. So we have a total of 4 phenotypes, which is A, B, AB and O, and 6 genotypes. Now, whenever somebody has A blood group, they could either be homozygous or heterozygous. Even in B blood group, they can be homozygous or heterozygous. So, when they have A blood group, their genotype can be IA or IA lowercase I. In B blood group also, they can be IB lowercase I. In both of these, you see normal dominance, like in Mendel. Okay, lowercase, in all three of them. Lowercase I, this is the recessive trait. Now, when it comes to B blood group, you have IA, IB. This shows codominance. Over here, I told you IA is responsible for the production of antigen A, IB is responsible for the production of antigen B. When in a dissimilar pair of alleles, both of these alleles IA and IB are present together, both of them will express such that on the surface of RBCs you have A antigen as well as B antigen. Okay, when they ask you which blood group shows codominance, not all blood groups do. It's only AB blood group in which you can see codominance. See here, if there is IAI, here also there is a dissimilar pair of alleles, here also there is dissimilar pair of alleles, heterozygous condition. But over here you see normal dominance, only one of the alleles are expressing. So among the three alleles, IA, IB, both of them are equivalent, both of them will express, and these are dominant to lowercase I. When this is present, when these alleles are present along with lowercase I, this will express, only one will express. But when these two are present together, both of them will express. Alright. Now, blood group is also example for another condition called multiple alleles. Generally for one character, for one gene, 2 alleles will be present. Whenever there is more than 2 alleles for the same gene, then we call it multiple alleles. Multiple means many, right? So whenever there is more than 2 alleles governing the same character, we call it multiple alleles. Here we have more than 2 alleles. There is one gene, we have 3 alleles, which is definitely more than 2. So these are the 3 alleles I've already told you. Blood group is an example for multiple alleles. AB blood group is an example for codominance. Alright? Yeah. Now, let's understand another thing. We say that a gene being dominant is not its autonomous feature. Okay. When we say a gene is dominant, it doesn't necessarily mean that let me put it this way. Generally, we see that one gene is responsible for one phenotypic effect, one trait. But sometimes it is possible that a single gene can control more than one phenotypic trait. Okay, I'll give you this example. In pea plant, the synthesis of starch is controlled by a gene called B. This B gene exists in 2 forms, capital B and lowercase b. Okay, so sometimes a single gene can have more than one effect. I'm giving you this example. In pea seeds, one gene controls the synthesis of starch. It has 2 alleles, capital B and lowercase b. Now, since it has 2 alleles, it can exist in 3 different genotypic combinations. It can be homozygous capital B, it can be heterozygous capital B, lowercase b, it can be homozygous lowercase b. Now, the single gene controls 2 phenotypic traits, 2 effects. What is the first one? The size of the starch grains. What is the second one? The shape of the seed. Okay, whenever it is in this combination, the alleles, starch synthesis will happen very, very efficiently. high efficiency you can see in starch synthesis. The size of the starch grains will be large. The shape of the seed will be round. Okay. Second, if it is heterozygous having a capital B lowercase p, of course it can synthesize starch grains, but efficiency will be moderate compared to capital B capital B. It will synthesize intermediate starch grains and the shape will be round, as you can see here. If it is lowercase p, efficiency of starch grain synthesis is the least among the three genotypes. Here, the starch grains are very small in size. As a result, the shape of the seed will be wrinkled. Now, it is one gene that is controlling this trait as well as this trait. If you look at size of starch grains, you have three phenotypes. Phenotype 1, either it's large or it's intermediate or it's small. If you see the shape of the seed, there are only two phenotypes. Either it is round or it is wrinkled. Now for the shape of the seed, the same gene is exhibiting dominance. If you consider the character size of the starch grains, the same gene is showing incomplete dominance. What happens in incomplete dominance? Homozygous dominant will show one phenotype, homozygous recessive one phenotype. When it is in heterozygous condition, it will be like neither of the parents, it will show intermediate, like in the case of snapdragon flower color. This is like normal Mendelian dominance. Whenever it is dissimilar, only one will dominate, only round will dominate. So you're seeing two phenotypes here. Okay. So if I say this gene is dominant, it is not always true, because that gene is responsible for two traits, and one is showing complete dominance and one is showing incomplete dominance. So you can't say that gene is always dominant, right? It depends on which character you're considering. If you're considering the character, color, shape of the seed, of course it is dominant. If you're considering size of starch grains, it's not showing complete dominance, it's showing incomplete dominance, right? So yeah. So we say that dominance is not an autonomous feature of a gene or the product that it has information for. Okay. Now here is a comparison between the three patterns of inheritance we studied so far. In dominance that Mendel explained, you have two phenotypes in the parental generation. Only one of them will express in the F1 generation. This will be heterozygous. It will show only one of the two traits. In incomplete dominance, it will show a brand new trait which is not present in either of the parent, but it will be intermediate. In codominance, you see a trait where both the parental traits are expressed. Okay, so simultaneously it will be expressed. If you have like a brown colored cow and a white colored bull and they are crossed, you will get a calf that has patches of brown and white where both are expressed. Okay, so these are the three different patterns of inheritance. Now let's understand the dihybrid cross. Mendel also worked, I told you, with two characters at a time. That is called two gene inheritance or a dihybrid cross. For example, what you're taking here is two characters of the seed. You know, for seed, two characters were there. One is the color of the seed, one is the shape of the seed. In color, yellow is dominant, green is recessive. In shape, round is dominant, wrinkled is recessive. So over here, we are taking one plant which has both dominant characters, both dominant traits. This is yellow and round, both are dominant. Here you have both recessive traits. Both of these are pure breeding varieties, true breeding varieties, which means they're both homozygous. This will be round and wrinkled. I will not explain this by writing again because you've already done this in monohybrid cross. I think you can follow. So you see here, we're starting with the parental generation. In the parental generation, you have round, yellow, wrinkled and green. We are writing the same genotype very similarly. Yellow is dominant, so we are using capital Y, capital Y. Green is recessive, so lowercase y. R is dominant, so we are going to use the first letter of round R. Round is dominant, so capital R, lowercase r. Why do we have four letters here? Because each character has two alleles. We have two characters here, so totally there will be four alleles. capital Y, capital Y, capital R crossed with lowercase y, lowercase r, lowercase r. In the F1 generation, you're going to get all plants that are yellow and round like that parent, and all of them will be heterozygous for both these characters. You'll have capital Y, lowercase y, capital R, lowercase r. What do we do next? We allow this plant, we allow that seed to germinate, and we will allow that plant to undergo self-pollination. After selfing, in the F2 generation, here are the different phenotypes you get. You basically get 4 different phenotypes. As you can see here, in this big triangle here, you have all yellow and round. If you see here, you have yellow and wrinkled. You have 9 yellow and round. You have 3 yellow and wrinkled. You have these 3 green and round. And you have this one green and wrinkled. If you notice here, we have new combinations. Yellow being wrinkled and green being round, we've not seen before. In the previous generations, all we've seen is yellow being round, green being wrinkled. But in the F2 generation, we have newer combinations, which means there is yellow that is being wrinkled, green that is being round. How do these newer combinations arise? We have to understand that. That is what Mendel also tried to understand. So here when you look at the ratio, you have 9 of these that are like this parent, yellow and round. In phenotype I'm telling, not genotype. You have 1 which is like this parent, which is wrinkled and green in phenotype. So you have 9 round and yellow, 1 wrinkled and green. These are like the parents, so we call it parental types. Total you have 9 plus 1, 10 parental types. These newer combinations are called non-parental types or recombinants. Okay, so you can see over here, this one, this one, this one, this one, this one, this one, a total of 6 of them are recombinants. Yellow and wrinkled 3, green and round there are 3. Alright, so we get the phenotypic ratio 9:3:3:1, where you have 9 round and yellow, 3 wrinkled and yellow, 3 green and round, 1 green and wrinkled. Okay, so this is the phenotypic ratio. Based on this result, what does Mendel propose? He proposes what is called the law of independent assortment. This is important to understand. Pay attention. Okay. So based on his observations, what he says is, whenever 2 pairs of traits, that means we're considering 2 characters, each character has 1 pair of alleles. So we're considering 2 pairs of traits in this dihybrid cross. Whenever 2 pairs of traits are combined in a hybrid, the segregation of 1 pair happens independently than the segregation of the other. Okay, so when 2 pairs of traits are combined in a hybrid, segregation of 1 pair of characters is independent of the other pair of characters. How can we say that? What he means is, yellow is not always, segregating with round. Green is always not segregating with wrinkled. Yellow, the character for color separation, segregation happens independently than the color segregation for shape. If both of them were linked, if both of them were dependent on each other, if both of them happened together at once, then there wouldn't have been possibility for these recombinants. The fact that we have recombinants here shows that yellow need not always be associated and dependent on round shape. Yellow can segregate either with round, yellow can segregate with wrinkled. So this character of color is not dependent on the character of shape of the seed. During gamete formation, these can segregate, these do segregate independently. Alright, that is what Mendel says in his third law, which is the law of independent assortment. This is based on dihybrid cross. So far we've learnt about 3 laws of Mendel. The first law is called the law of dominance. Law of dominance doesn't hold true for all organisms. We saw how it is not true in the case of in the case of incomplete dominance and codominance. Law of segregation is a universal law. It is applicable to every organism diploid sexually reproducing. Law of segregation basically said that when diploid organisms reproduce, only one set of chromosomes get passed on to the uh, next generation through gametes, right? So it gets segregated. That is what it said. It is universal for diploid sexually reproducing organisms. Talking about Mendel's dihybrid cross, the law that he proposed based on Mendel's dihybrid cross, which is the law of independent assortment, this is also not true for all living organisms, for all genes. Why it is not true, I'll explain it going forward. Okay. I mentioned how we call Mendel Mendel the father of genetics and all of that, but Mendel doesn't really have any idea that he is so celebrated in the field of genetics because his work wasn't appreciated when it was published. After he passed away, people rediscovered his work and then he was called the father of genetics. But for as long as he was alive, he didn't know that his work was so significant. He published his work on inheritance in the year 1865. Okay. It was recognized until 1900 for a few reasons. Firstly, it was not like today's times where you post something on Instagram and it goes viral within a few hours, right? Communication was a problem. So it could not be publicized very widely, first of all. Secondly, he said that genes or factors, as he called them, are discrete units. They don't change. They are as it is passed on from the parents to the offspring via the gametes. Many people did not accept that because in nature you see so much variation. If the genes or alleles or factors are passed on as they are, then how is it that there is so much variation in nature is something that many people could not comprehend. Okay. And thirdly, he used mathematics, like I mentioned earlier, to explain biological phenomena, which is very, very new at that time and not people accepted it. And finally, it was because he said that there is something called factor that is getting passed on from one generation to another, but he couldn't provide any physical proof for it. He couldn't tell that factors exist inside of cells, they are present in chromosomes, they are just basically DNA. He could not provide like a physical proof for the existence of factors inside our bodies. And these are some of the reasons why his work remained undiscovered and unaccepted and not so publicized for a really long period of time. See, it was published in 1865. At that time, nobody appreciated it. Later in the year 1900, three people independently rediscovered his work. See, whenever somebody is performing any new research on something, it's always advised that you go through the work that has been done in a similar field so that you get an idea of what are the different approaches people have taken for research. So you do like a study of the work that people have done before you to get an idea of what you could do. So while these people were also interested and curious about genetics and wanted to learn how inheritance works, they studied whatever previously researchers had worked and they discovered his work. And these three people are Hugo de Vries, Carl Correns and Schermak, Erich von Schermak. They weren't working together, they independently rediscovered his work. Okay? Yeah. So by then, around the time 1900, microscopy was very advanced and people had worked out how the chromosomes behave during the process of meiosis. When does meiosis take place? During gamete formation, during gametogenesis, right? So during gamete formation, meiosis takes place. During meiosis, how are the chromosomes behaving? They are coming and aligning at the center. They are getting segregated. They are getting separated. Chromatids are pulled apart during meiosis too. So all of these they had worked out, okay? And the knowledge they had from the behavior and movement of chromosomes during meiosis, they tried to apply that to what Mendel had said. And they came up with something called chromosomal theory of inheritance. This was proposed by two people, Sutton and Boveri. Okay, so they united the information about how chromosomes move and behave during meiotic cell division, and they applied that to the principles and laws that Mendel had proposed. Okay, and they noted how the behavior of chromosomes during meiosis was very similar to how genes or factors behaved. Whatever Mendel said about factors, it was very, very similar. Okay, and that is called the chromosomal theory of inheritance. You know about meiosis. During cell division, meiotic cell division, 2 cycles will take place, and diploid cells will give rise to haploid gametes. We know all of that, right? So, what do we know about chromosomes is given in these green boxes. You can see how similar to factors, chromosomes also occur in pairs. Similar to how Mendel said factors segregate during gamete formation, we see chromosomes also segregate during gamete formation. Similar to how Mendel said factors segregate independent of each other, we see that chromosomes also segregate independent of each other. So ultimately they said that whatever factors or genes Mendel is talking about, they are nothing but they are present on the chromosomes itself. Alright, so they united the knowledge of chromosomal segregation with the principles of Mendel, and they called it chromosomal theory of inheritance. Okay? Yeah. Now, in order to prove experimentally the chromosomal theory of inheritance, Thomas Hunt Morgan conducted some experiments. Okay? So, this is Thomas Hunt Morgan. He experimentally verified the chromosomal theory of inheritance. He worked on an animal, unlike Mendel, and the animal that he chose was fruit fly, whose scientific name is Drosophila melanogaster, tiny flies that you see around fruits, bananas especially, if they are kind of like ripe, or even you can see it around curd and all of that. Okay. So again, why did he choose Drosophila melanogaster? For the same reasons that Mendel chose P. It can be grown very easily on a very simple synthetic medium, not much effort there, not much hassle. And they also complete their life cycle in around 2 weeks itself. Okay, so they have a short lifespan and a life cycle. After just one mating, they produce many progeny flies, like how peas produce so many seeds. Here there was a very clear distinction between sexes. The males and females could be very easily distinguished, and they also had a lot of characters with opposing traits that was easily observable with a new low power microscope itself. So that is the reason he chose the fruit fly. And what did he do? He performed the dihybrid cross similar to what Mendel did. Here he chooses 2 characters. Of course, it's a dihybrid cross. 2 characters have to be chosen. The characters that he chooses are both related to color. One is color of the body, the other is color of the eyes. Body color exists in 2 forms. Either it can be yellow or it can be brown. Eye color, either it is white or it is red. Over here, yellow is dominant, yellow is recessive, white is recessive, brown is dominant, red is dominant. He calls the dominant varieties the wild type and the recessive varieties he calls as mutants. While representing this in crosses, generally what would we do in Mendel's experiments? You would take whichever is dominant, use that first letter of that trait. You would have chosen brown to be capital B, yellow to be lowercase b, but here that is not what he does. Yellow is represented as Y, lowercase y. We are taking the first letter of the recessive trait. For brown, you will write Y with a superscript plus. White will be W, red will be W plus. Okay, so both of these characters were situated on the X chromosome. In Drosophila, similar to how in humans we have females having 2 X chromosomes and males having 1 X and Y chromosome, very similarly even in Drosophila, females will have 2 X chromosomes, males will have 1 X and 1 Y. So what does he do in the parental generation? he chooses the female to be recessive or mutant in both the characters, and he chooses the male to be dominant or the wild type for both the characters. What you get here in the offspring, that is the F1 generation. Here you cannot self-pollinate it because it's an animal. But among the progeny, whatever males and females you get, you cross them. That is called intercrossing. From that we will get the F2 generation. Now let's see. We start off with the first parental generation. Female has 2 X chromosomes represented by pink color bars. Male has 1 X and 1 Y represented by blue color. I told you female is recessive, so she will have YW, that is the alleles. And the male is dominant or wild type, brown body, red eye, so you have Y plus W plus. I want you to notice how, since the female has 2 X chromosomes, she has 2 copies for each character. She has 2 alleles. Males have only 1 X chromosome, so they have only 1 copy. Since female is homozygous, she will produce just one type of gamete. That gamete will contain the same allele Y and W, both recessive. Since males are, here we don't call them heterozygous, we call them hemizygous. That means they have only one copy of each allele. Because they have not dissimilar pair of alleles, they have 2 different types of sex chromosomes. So here, since you have 2 different types of sex chromosomes, 2 types of gametes can be formed. In one subset of gametes, one subset of sperms, X chromosome will be passed on. In the other subset of sperms, the Y chromosome will be passed on. Now, if you plot this in a Punnett square, we will get females that are like the father, we will get males that are like the mother. Okay? So, the female will be showing, she will get this X chromosome from her father, which has the dominant alleles. She will get an X chromosome from the mother, which has the recessive alleles. So this female is heterozygous dominant. The male has only 1 X chromosome, so whatever is present there, that only has to express. He has received the X chromosome from the female, from his mother, and that has both recessive alleles. So this male will be yellow-bodied and white-eyed. Okay, so this is our F1 generation. What are we going to do now? We're going to cross these two, intercross them. Okay, so then we'll get the F2 generation. So after intercrossing F1, what do we have? We have a female showing the dominant phenotype, heterozygous condition. We have a male showing the recessive phenotype, hemizygous. Since this is heterozygous, there are 2 possibilities for gametes. Since this is hemizygous, you have 2 possibilities for gametes. Over here, some of the gametes, this X chromosome will go. To some of the gametes, this X chromosome will go. Over here, to some of the gametes, X chromosome will go. To one subset, Y chromosome will go. Again, plot this on a Punnett square. We are going to get these combinations. Here are the females. In females, you have yellow body, white eye, brown body, red eye also. And in males, you have yellow body, white eye, brown body, red eyes also. So all of these that you're looking at here are the parental types. What were the parental types? Yellow body with white eyes, brown body with red eyes, right? So you get these parental types. Now, you know that during gamete formation or meiosis, something called crossing over takes place. During crossing over, homologous chromosomes come close to each other and segments of chromosomes will get exchanged between the two during the Pachytene stage, right? So this is what happens. This you've learned about in the chapter cell cycle and cell division. So when these homologous chromosomes come in synapse, segments will get exchanged such that each chromosome becomes a mixture of both the parental chromosomes. Since even over here, during gamete formation, meiosis happens, there are chances that when the 2 X chromosomes, remember here crossing over can happen only in females, not in males, because males don't have homologous chromosomes. They have 2 separate chromosomes, X and Y. Between X and Y, crossing over is not possible. Crossing over can happen between X and X only. Now in this case, let's say, because this female here that we took, this female is heterozygous, right? So if you consider this female during gamete formation, there are chances of crossing over, and when crossing over happens, let's say this segment of chromosome got exchanged. Initially there was Y with W, Y plus with W plus. If crossing over has happened, you will have Y plus with W, Y with W plus, and newer combinations have formed. If newer combinations have formed, then her gametes are going to be like this, half pink, half blue mixture, because recombination has occurred. No change in male, same gametes will be present. If you look here, you will get recombinants. You will get females that have brown body with white eyes. Initially it was brown body with red eye in parental. Now you have a newer combination. Brown body with white eye. You have yellow body with red eye. Likewise, in males also you have brown body, white eye, yellow body, red eye. These are the recombinants. According to Mendel, in his dihybrid cross results, he obtained 6 out of 16 as recombinants, 10 out of 16 as parental types. Right? This is Mendel's dihybrid cross. In that case, recombinant would be 37.5%. Parental type would be the remaining, which is 62.5%. Right? But when Morgan performed this experiment, he saw that recombinants were only 1.3%. Parental types were 98.7%, which was a huge deviation from Mendel's dihybrid cross phenotypic ratio. He performs another monohybrid cross, another dihybrid cross using fruit flies itself, using 2 other characters. This time he chooses the color of the eyes, eye color, and wing shape or wing type, if the wing is normal or miniature. When he performs a dihybrid cross using these 2 characters, he will notice that the recombinants are somewhere similar to Mendel's dihybrid cross, 37.2%, 62.8%. Okay, so we saw here, right, previously, in Mendel's dihybrid cross, we got 37.5, 62.5. And here we got 37.2, 62.8, which is kind of close. So he tries to understand if genes are segregating independent of each other, then why am I not getting the same ratio in this case? Why is there such a huge deviation? I got barely any recombinants. I got mostly parental types, just 1.3% recombinants. And then he would know that all of these genes that he is considering are present on the X chromosome. And based on his observation, he says that the 2 genes in the first cross that we spoke about did not segregate independently because they are present on the same chromosome. Okay, so F2 ratio deviated very significantly from this. In the parental type, normally you would get 62.5% in Mendel's cross, recombinants were 37.5. But in the first cross, we saw a huge deviation. In the second cross also there was a deviation, but not as much. Okay, so he says that they did not segregate independently. What he already knew was that the genes were located on the X chromosome. Whenever 2 genes are located on the same chromosome and you're performing a dihybrid cross using those 2 characters, how many recombinants you get depends on how close they are to each other or the distance between them. If the 2 genes distance is large, if the distance between them is more, then the number of recombinants you get also will be more. If the 2 genes are present very close to each other, then the number of recombinants that you will get will also be less. Okay, so I want you to notice here, in the second cross he got more recombinants. The distance between these 2 genes is more. In the first cross, he did not get many recombinants because the distance between them is close. Because they're present so close to each other, crossing over does not happen at gene level. It is probable that this whole thing got exchanged so that we did not get any new recombinant at all. What was YW here got exchanged with Y plus W plus. Because that happened, not there. Over here, since this got exchanged as a segment from here to here, we did not get newer combinations. Newer combinations possibility is more when they are farther away from each other, right? So the distance between the two genes we are considering plays an important role when we are performing a dihybrid cross, considering genes present on the same chromosome. We say that all the genes present on a chromosome are linked. If they are present close to each other, they are tightly linked. If they are present a little far away from each other, then they are loosely linked. The more tightly the genes are linked, the lesser the distance between the two, the chances of getting recombinants is also less. If the distance between them is high, then the chances of getting recombinants is also high. Okay? So here, he coins two terms, linkage and recombination. Linkage is a term that is used to describe the physical association of genes on a chromosome. If there are genes on a chromosome, then all of that DNA is connected. They are physically attached to each other, right? They are physically associated with each other. That is linkage. What is recombination? Recombination is the generation of non-parental gene combinations. Whatever newer combinations we get, to describe that term, he uses recombination. Now, what do we infer from this? Even when genes are grouped on the same chromosome, some of them are very tightly linked. They showed lower recombinants or non-parental types. Whenever genes are loosely linked, they showed more recombinants. Okay, so example for when they were tightly linked, the first experiment, eye color and body color. The second time when they considered eye color and wing shape, they were located on the same chromosome but they were distant. So you got more recombinants. Okay, so that is Thomas Hunt Morgan's experiment. Morgan had a student in his lab. His name is Alfred Strutewert. What he did was, he checked for how many recombinants he was getting. Based on the frequency of recombinants, which we call the recombination frequency, he started to develop genetic maps. See, I know that if the recombination is this much, then if it is less, then the two genes that I'm considering are close to each other. If the recombination is this much, then the two genes that I'm considering are farther away from each other. Okay, so this we know. Depending upon, based on how much recombination we get, you can kind of predict whether the two genes are close to each other or present far away from each other. Based on that, he started to construct genetic maps. Okay, based on the distance present between the genes, he mapped them, and that is used as a starting point in many projects like the Human Genome Project, about which you'll learn in the next chapter. Now let's learn about a concept called polygenic inheritance. In whatever experiments Mendel performed, he considered characters that had distinct alternate forms, two distinct alternate forms. But in nature, every character will not exist in only two starkly distinct contrasting features, right? There are many characters in nature which are spread across a gradient. Like if you think about height in humans, it's not like a person can either be 6 feet tall or 5 feet tall. There is a gradient. Some people are 6, some people are 5'8", some people are 5'9", I am 5'3", you could be 5'7", whatever. Some people could be 5'2", or even shorter. There is a gradient. If you think about skin color, there is a gradient. It's not like black and white. There's black, like very dark, like brown, like most of us, and then there's very pale. So you have a gradient in skin color. So for examples, you have height and also skin color. Such characters which appear in the form of a gradient are generally thought to be controlled by more than two genes. Minimum 3 or more. Okay, I don't want you to get confused between multiple alleles and polygenic inheritance. Multiple alleles is when one gene has more than 2 alternating forms. Polygenic inheritance is when one character is controlled by more than 2 genes. Alright. So generally polygenic inheritance takes into account the influence of environment as well. For example, if you think about height, we all have genes that are responsible for height. Probably more than 2 genes are present. I can, according to my genes, grow to a height of 5 feet, let's say 6 inches, but I don't eat nutritious food. I don't exercise well enough. Then even though I have the genes that can allow me to grow that tall, because of my poor nutrition and exercise, I may not grow that much. According to the genes that I have for skin color, probably I should be light brown. But I'm spending all day playing cricket in the sun. Obviously, I will turn darker, right? It will take into account the environment's influence as well. Okay, so that is polygenic inheritance. Let's take example of human skin color. Let's assume the human skin color is controlled by 3 different genes, A, B and C. It can exist in dominant or recessive form. Capital ABC is dominant, lowercase abc is recessive. Now I want you to understand how polygenic inheritance is quantitative or additive. The contribution of each allele will matter. If you have more dominant alleles, that's going to be the darkest skin color. If you have the least dominant alleles or all recessive alleles, then that is going to be like the palest skin color. Here it doesn't matter whether A is dominant, B is dominant or C is dominant. What matters is how many alleles are dominant. Okay, for example, let's see here. If all of them are dominant, it's going to be the darkest skin color. If all of them are recessive, it's going to be lightest skin color. If 5 of them are dominant, it's going to be slightly lighter than all 6 dominant. Now over here, 5 dominant alleles could be A, B, B, C, C or A, B, B, C, C or A, B, B, C, C. Okay. In all these 3 genotypic combinations, you can see there is one recessive allele and remaining dominant alleles. So in all of this will be the phenotype. It doesn't matter which one is dominant. What matters is how many are dominant. Likewise, if 2 are dominant, as you can see, the fewer the dominant alleles, the more paler the skin color will be. Okay. So it is additive. More dominant alleles, darker will be the skin color. Okay. That is polygenic inheritance. Then we have pleiotropy. Pleiotropy is a phenomenon where one single gene controls more than one phenotypic trait. Okay. So a single gene responsible for multiple phenotypic expressions is called pleiotropy. One character controlled by more than 2 genes is polygenic inheritance. This is kind of like the opposite. One gene controlling more than one phenotypic expression is pleiotropy. Okay. So, example we can take phenylketonuria, which is a disorder. Okay, what happens here is there is one gene. That gene is responsible for production of an enzyme called phenylalanine hydroxylase. Now that gene gets mutated. PAH, phenylalanine hydroxylase, is not getting produced. Okay, because that one gene is affected, one gene is not functioning normally. Multiple phenotypic effects you can see. What are the multiple phenotypic effects? In such individuals who suffer from phenylketonuria, the pigment in the skin is reduced. There is reduction of pigment in the hair and reduction of hair also. You can see mental retardation in such individuals. So these are distinct phenotypic expressions that are caused because of a single gene. So this is called pleiotropy. More about what happens exactly during phenylketonuria we will discuss when we talk about genetic disorders. But for now, understand that with respect to pleiotropy, phenylketonuria is used as an example. One gene is affected. Phenylalanine hydroxylase it's supposed to produce, because it's mutated, it's not producing that enzyme. Because of that mutated gene, mutated allele being present, all of these phenotypic expressions are seen. Okay, so one gene affecting multiple phenotypic expressions is what we call pleiotropy. Now, let's understand sex determination. You know how sex determination happens in humans, right? In humans, males have 44 autosomes, normal males, and an X and a Y chromosome, and the females have 44 autosomes plus 2 X chromosomes. These males undergo meiotic cell division during the formation of the male gamete sperm. Since they are hemizygous, they have two different types of sex chromosomes, they will produce two types of gametes. Okay, one subset of sperms will contain half the number means 22 autosomes plus X. The other subset of sperms will contain 22 autosomes plus Y. In females, when they undergo meiotic cell division, they will all produce same type of egg, which has 22 autosomes plus X chromosome. So these cells are haploid, you know that. When the sperm containing X chromosome fuses with the egg containing X chromosome, you get 44 autosomes with XX, and this will become a female child. And if the sperm containing Y chromosome fuses with the egg containing X chromosome, it will result in 44 autosomes plus XY, which will become a male child. Since here males produce two different types of gametes because they have two different types of sex chromosomes, males are said to be heterogametic. Hetero means different. Different types of gametes it's producing, so it's called heterogametic. Since females have only one type of sex chromosome, females produce only one type of gamete, females are said to be homogametic. Okay, now let's understand how sex determination will happen in some other organisms. If sex determination, it can happen in various ways, either because of the environmental influence or because of sex chromosomes. Here we'll see some other examples of how sex determination happens because of sex chromosomes. Okay, we saw how sex determination happens in us humans, right? In humans, males are heterogametic, females are homogametic, very similar to how sex determination happens in us humans. It happens in Drosophila as well. In Drosophila, the females have 2 X chromosomes and the males have 1 X and 1 Y chromosome. Even over here, males are heterogametic. Males will produce two different types of gametes. Females will produce only one gamete, all containing X chromosome. Males will produce 50% sperms containing X, 50% sperms containing Y. Likewise, we have grasshopper, which exhibits male heterogamety. Unlike what we saw in humans and Drosophila, over here there is only one type of sex chromosome. But there is a difference in the number of sex chromosomes. While females contain 2 X chromosomes like us, the males here do not contain 1 X and 1 Y, they contain only 1 X. What is O here then? O means nothing, 0, equivalent to 0. Males will have XO chromosomes. Okay, so here in males, one subset of sperms will contain X chromosomes, the other subset of sperms will not contain any sex chromosome. Okay, if the sperm containing no sex chromosomes fuses with the egg, it will become a male. If the sperm containing X chromosome fuses with the egg, it will become a female. Okay. Heterogamety need not be restricted only to males. You can see that in females also, in birds and butterflies. Whenever there is female heterogamety, we don't use the letter X and Y, we use the term Z and W. ZW type of heterogamety you can see in birds. Over here, males will have same sex chromosome, 2 Z chromosomes. But females will have 1 Z and 1 W. Some of her eggs will be Z, some of her eggs will be W. So if the sperm fuses with the egg containing Z chromosome, it will become a male. If the sperm fertilizes an egg containing W chromosome, it will become the female. In butterflies, similar to grasshopper, you can see ZZZO type of ZOZZ type of sex determination. Over here, there's only one type of sex chromosome, Z chromosome. Males have 2 copies of them, females have only 1 copy of them. So, in some eggs, you will have Z chromosome, in some eggs, you will not have any sex chromosome. If a sperm containing Z chromosome fuses with the egg containing Z chromosome, it will become a male. If a sperm containing Z chromosome fuses with the egg containing no sex chromosome, it will become a female. Since females in these cases produce 2 different types of gametes or 2 different types of eggs, we say this is female heterogamety. Now this is very interesting how sex determination happens in honeybees. In honeybees, the type of sex determination is called haplodiploidy. Over here, the females are diploid containing 32 chromosomes. Males are haploid containing 16 chromosomes. The female, especially in bees, the queen is the only fertile female. Only queen reproduces, workers do not reproduce, they are sterile. So the female queen will undergo meiosis to produce haploid egg cell containing 16 chromosomes. The male is already haploid. Male will not undergo meiosis. Male will undergo mitosis to produce sperms that contain 16 chromosomes. Now, this queen can fertilize her egg using the sperm from the drone and get a zygote with 32 chromosomes. This will become females. If she does not choose to fertilize her egg with the sperm, it will parthenogenetically develop into males that are haploid. So over here the sex that depends not on which sex chromosome you have, but on how many sets of chromosomes you have. If they are diploid, they are females. If they are haploid, they are males. Generally we say during gamete formation meiosis takes place, but here it is unusual since the males are haploid themselves. During sperm formation, during gametogenesis, they will not undergo meiosis, they will instead undergo mitosis, and the sperms will form after mitosis as a result of mitosis, and females undergo meiosis to produce the egg. The queen can choose to fertilize her egg if she wants more workers in the hive, and the queen, if she wants more drones in the hive, she can allow the egg to parthenogenetically develop into the male. Okay, so I want you to understand how the males here don't have fathers at all because they directly develop from the egg. They cannot have sons at all because for the formation of male, sperm is not required. So drones cannot have sons, they don't have fathers, but they have grandfathers. This female would have had a father, no? The queen would have had a father. So through his mother, there is a grandfather, and through his daughter, he can have a grandson. So drones don't have fathers, they don't have sons, but they have grandfathers, they can have grandsons. Okay, so yeah, that's about sex determination. Now let's talk about mutation. I've told you roughly what mutation is. Whenever there is any change in the sequence of nucleotides in DNA, we refer to it as mutation. Mutation will bring about variation in DNA. It can result in the formation of new alleles for a gene. How does this mutation happen? It can happen because of various reasons. Mutation can happen at a nucleotide level or it can happen at a chromosome level also. In a chromosome, some segment can get detached, go and attach to some other chromosome. It can get deleted, it can get translocated from one chromosome to another. All of that is possible at the chromosome level also, at the DNA level also. What causes these mutations? There are certain physical agents and chemical agents. Certain biological agents also can cause mutation, like some viruses. Example for physical agents, like I've told you earlier, some wavelengths in the sun's radiation, certain types of UV can cause mutation. The smoke in cigarette, tobacco, that can cause mutation. Whatever factors cause mutation, they are referred to as mutagens. Now let's talk about pedigree analysis. You know how many characters get transferred from one generation to another. If you have to study plants or some other animals, you can perform crosses and find out how inheritance of that particular character or that particular trait is happening. But if it's in humans, it's difficult to perform crosses like that because it's not morally correct. And if you have to analyze any trait over several generations, the easier thing to do is something called pedigree analysis. So using this, we can trace how exactly inheritance is happening for a particular trait. It need not be a disease, it could be any trait, any abnormality, any disease, something. In this pedigree analysis, we use some symbols. Okay, we'll understand those symbols. To represent males, we use a square. To represent female, we use a circle. If sometimes you don't know in some previous generation whether it was a male or a female, if you're not really sure, it will be diamond shape, which is sex unspecified, rhombus. If the female or male or sex unspecified is affected or has the trait which you're studying, you will shade them entirely. Marriage is indicated by a line between a circle and a square. If there is consanguineous mating or marriage that has happened between very close relatives like first cousins, you will represent that by two lines. Why is that important? Because you can see many times when there are closely related individuals getting married and having children, they are more prone to having like chromosomal or genetic disorders, or you can see like deafness, being dumb, or mental retardation in such individuals. Okay, yeah. And here you can see this man got married to this woman. They have two children. It is represented like this in the order of the childbirth. First was a girl, then was a boy. That's why it's represented like this. Again, if the child is affected, we have to shade. If you know that one couple had these many children, but you're not really sure which is a boy, which is girl, you just write it like this and you write the number of children there. Okay. All right. Now we have to learn about genetic disorders. Genetic disorders are of two types. You have Mendelian disorders and chromosomal disorders. Mendelian disorders are disorders that are passed on from one generation to another, similar to how Mendel said characters pass on. Chromosomal disorders happen because of aberrations or mutations that have happened at the chromosomal level. Generally, Mendelian disorders are formed as a result of just mutation in one gene. Chromosomal disorders, at the chromosomal level, there will be some abnormality. Again, the gene that is responsible for the disorder in Mendelian disorders could be located on the sex chromosome or on the autosomes. If it's located on the sex chromosome, we call it sex-linked. If it is located on the autosome, we call it autosomal. Again, that disorder could express itself, manifest itself in its dominant condition or in its recessive condition. If it expresses in its dominant condition, we call it sex-linked dominant or autosomal dominant. If it expresses in recessive condition, then we call it sex-linked recessive or autosomal recessive. So chromosomal disorders, we learn about that also. Okay. Now, pedigree analysis you can use to understand whether any disorder or any trait that you're studying is dominant or recessive. Usually to analyze Mendelian disorders, you can use the pedigree chart. Over here, example is given of myotonic dystrophy. This is an autosomal dominant trait. Whenever there is an autosomal dominant trait, you can see how the parents, unless the parents are affected, the offspring will not be affected. Over here you can see among this couple, the lady is affected. So some of her children are affected. Okay, again here you see the lady is affected, then one of her daughters or her daughter is affected. So in autosomal dominant traits, you see the trait will express itself in the next generation only if it has been expressed in the previous generation as well. Okay, So here it is expressed, that is why it is expressed here as well. See, this lady in autosomal dominant trait, what happens is, let's say A, these are the combinations. If these two combinations are there, they can see disease. If this combination is there, then they'll be normal. So this person will be lowercase a. Now this lady can be capital A or capital A lowercase a. How do we find out whether she is this or this? Look at her children. If she was capital A, then every one of her children would have received that defective allele from her. Even if they have one copy of that defective allele, they would have shown the symptoms of disease. But over here, that is not the case. Some of them are showing the disease trait, some of them are not, which means she is not this, she is this. So in these children, she has passed on the capital A allele. They've received lowercase a from the father. But in the children that are normal, she has passed on lowercase a. Again, same thing here. This individual could be heterozygous. These both are homozygous recessive. Understood? That is how autosomal dominant traits work. Now, how autosomal recessive trait, for example, sickle cell anemia, is different from autosomal dominant trait is, over here, since it is recessive, even if the parents are affected, you can see that the offspring can be affected even if the parents are not showing symptoms of the disease. Over here, capital A, lowercase a are normal. Lowercase a shows the disease. Okay. So here parents are appearing normal. This is lowercase a. If they have received lowercase a one from each parent, then obviously the parents should have one copy of the defective allele, right? But they are not themselves showing the symptoms, which means they will have one normal allele also. For the children that are not showing the symptoms of that disease, at least one normal allele will be passed on from one of the parents. Unfortunately, for these two children, this boy and this girl, both of them have passed on the defective allele. That is why they are suffering from that disorder. Okay, so in this generation also you can see these two parents are not showing any symptoms of the disease. But this boy here is showing the symptoms of disease, which means he has lowercase a. If he has two copies of lowercase a, obviously each of his parents will have one copy. That is how he can get it. But they themselves are not showing the symptoms of disease, which means they are capital A, lowercase a. Okay, what we need to understand here basically is when we say it's an autosomal dominant trait, the dominant allele is the defective allele that is causing the disease. If there is recessive allele, then that will be normal. Okay. So even if there's one copy of the defective allele, even in heterozygous condition, it is going to show the symptoms of disease. But in case of autosomal recessive traits, this capital A allele is normal and lowercase a is recessive. You know lowercase a recessive is the defective allele. You know that recessive traits express only in homozygous condition. So unless there is two copies of the defective allele, you will not see the symptoms of the disease. In dominant disorders, if you see parents are affected, at least one of the parents are showing the symptoms of the disease or showing the trait, then only the offspring will show the trait. But if the parents are not showing the trait and you see the trait in the offspring, in the children, then it is recessive. Okay? Now, there are some Mendelian disorders we have to study. We'll start with color blindness. Color blindness is an X-linked recessive disorder. So what basically happens in color blindness is not that people will see black and white. They can see color. You know, the color vision, we can see color because of certain photoreceptors present in the retina of our eye. We have rods and cones, two different types. Cones are what is responsible for colored vision. Rods are responsible for twilight vision. So you have different types of cones. The cones that are responsible for showing us, separating out red and green color, the genes responsible for that would have gotten mutated, due to which they can't really differentiate between red and green colors. Okay. So the gene that is responsible for that is present on the X chromosome. Since X chromosome is a sex chromosome, this disorder is called autosomal recessive disorder. Okay. Now, if you look at the frequency of occurrence of color blindness in a human population, it is more common in males than in females. In males, among males, about 8% of them will show color blindness, while only about 0.4% of females show color blindness. Why is that? Females have 2 X chromosomes, twice the number of X chromosomes. Then would it not be that the females show more color blindness than males? Not really. I'll tell you why. See, females have 2 X chromosomes, males have 1 X and 1 Y, right? So in color blindness, let's say this is the normal allele, X is normal allele, and XC is the defective allele that causes color blindness. X with a superscript C is the defective allele that has color blindness. Now in female, there are 3 possibilities. Either the female is XX, or the female has 1 normal allele, 1 defective allele, or the female has both defective alleles. I told you that this is an X-linked recessive disorder. The normal allele here is dominant. The defective allele here is recessive. So if a lady has to be colorblind, then she should have 2 copies of the recessive allele, because recessive traits don't express themselves unless they are in homozygous condition. So whichever female has this combination of alleles in her X chromosome will show color blindness. Both of her alleles are mutated, so there is no distinction between red and green colors. This woman has both normal alleles, so obviously she is normal. What about this woman? She has 1 normal allele, 1 defective allele. Because of the presence of a defective allele, there could be some cones that are not normal. But she also has a normal allele which will make up for it. Because of the presence of this normal allele, some cones will be proper, right? With just those, she will have the ability to distinguish between color. So she herself is not color blind. She will be normal, but she has a defective allele. She is carrying a defective allele which she can pass on to her next generation. So we call such heterozygous individuals carriers. Talking about males. Males have an X and a Y. There is only 2 possibilities. Either they have normal XY or they have defective allele X and Y. So if a male has this combination of allele, he will be normal. If this is the case, then he will be colorblind. If a man has just 1 defective allele, that is sufficient for him to be colorblind. Because males and humans are hemizygous, they have only 1 copy of X chromosome. Whatever is there on that X chromosome, if it's normal or recessive, that itself will express, because they don't have another X. So even if a man has 1 copy of X chromosome, he will defective allele on the X chromosome, he will show color blindness, whereas that's not the case for women. If they have just 1 copy, they will not be colorblind. Of course, they will have a defective allele, they will be carriers, but they themselves will not be colorblind. For a woman to be colorblind, both her X chromosomes should carry the defective alleles. Now, when is that possible? We know about sex determination. If a female child has to form, she gets 1 X chromosome from the mother through her egg and 1 X chromosome from her father through the sperm, right? let's say this carries X. Only when these two come together, you will have a child that is born with 2 X chromosomes, which is a girl. Now this female, if she has to be colorblind, she gets 1 X chromosome from her father, 1 X chromosome from her mother. Father has only 1 X chromosome, right? So if she has to be colorblind, she should have both defective alleles. If she has both defective alleles, she has received one allele from her father, then her father 100% must be colorblind. If a daughter is showing symptoms of colorblindness, then 100% her father also will be colorblind. And her mother is at least a carrier, or she also can be colorblind. Father has to be colorblind, mother at least a carrier. Two colorblind people getting married to each other, or one carrier and one colorblind getting married, is not very common, right? If you think about how it occurs in males, males have 1 X, 1 Y chromosome. They receive the X chromosome from the mother, Y chromosome from the father. So there's no way that a man with X and Y chromosome can transmit that disorder, colorblindness, to the son. If a male is colorblind, then he would have gotten colorblindness from the mother only, because mother only passes on the X chromosome and colorblindness related gene is present on the X chromosome itself. A man cannot get colorblindness from his father. He gets it from his mother only. Okay. So for this lady to pass on colorblindness to her son, it's not necessary that she has to be a colorblind woman. Even if she is a carrier, there is still a possibility. Just a carrier woman can pass on colorblindness allele or colorblindness to her son. But for a woman or a girl child to develop colorblindness, father should 100% be colorblind and mother should at least be a carrier. Since this is the requirement for females to be colorblind, and the chances are rare. Okay, so it's more commonly seen in boys than in females. Alright, so males have only 1 X chromosome, females have 2 of them. The son of a woman who carries the gene has 50% chance of becoming colorblind. If she is a carrier, like I told you here, there are 50% chances of her passing on the defective allele to her son and 50% chances of her passing on the normal allele to her son. There is a 50% probability even if just the mother is a carrier, right? And yeah, if mother herself is not colorblind, because the gene is recessive, it expresses only in homozygous condition. It means that the effect is suppressed by her dominant allele. I already told you this. A daughter will not normally be colorblind unless her mother is a carrier and her father is colorblind. This is how X-linked recessive disorders get transmitted. So they call this crisscross inheritance also, because daughter gets it from the father, sons get it from the mother, right? Now let's understand hemophilia. Hemophilia is also something very similar. This is also a sex-linked disorder which is recessive. Here also we are calling this sex-linked recessive disorder because the defective gene is present on X chromosome. Same story again, males have 1 chromosome, 1 X chromosome only and 1 Y, females have 2 X chromosomes. What basically happens in hemophilia is, it is a blood related disorder. You've learnt in the chapter body fluid and circulation how clotting of blood happens, right? Whenever there's a wound or a cut, blood starts oozing out and then eventually it will stop, a clot will form. For the formation of clot, a lot of different things are required. Platelets are required, a whole different set of proteins are required. Like that, one clotting factor, one protein is also required. Many clotting factors are required. One clotting factor which is required in the process of clotting, that clotting factor is not formed because that gene is mutated. Okay. Because of the absence of just one clotting factor that is involved in blood clotting, in such people who have hemophilia, blood will not stop, bleeding will not stop at all. They could bleed sometimes if they are not assisted immediately to death. Okay. So Here also transmission happens from a carrier female who is not showing the symptoms of the disorder to her sons. Okay, here there is a single protein that is affected. It is a part of a cascade of proteins that are involved in blood clotting. Because of this, a simple cut could result in non-stop bleeding. Here again, the heterozygous female carrier for hemophilia may transmit the disease to the sons. A female becoming hemophilic is extremely rare because mother of such a female has to be at least a carrier and the father has to be hemophilic, like we saw in color blindness. What happens in such cases is, whenever a man who has hemophilia marries a carrier woman, in the embryonic stages itself, the embryo will not survive. Okay, so the child itself may not form. So it's very rare. The embryo will not be viable in the later stages of life. This is also called the royal disease because Queen Victoria, you know Queen Victoria, right? In her family, many people apparently suffered from this disease. So it is also called the royal disease. Now, let's talk about sickle cell anemia. Just 1 minute, let me rearrange this. All right. Okay, we'll finish phenylketonuria, then we'll talk about sickle cell anemia. Phenylketonuria, I've already discussed this as an example for pleiotropy. I told you there is one gene that is affected. That is the gene that is responsible for the formation of phenylalanine hydroxylase, an enzyme. So what happens basically in phenylketonuria is that one gene is affected. That one enzyme PAH is not produced. What is the function of PAH? It is an enzyme that is involved in the metabolism of amino acids. Phenylalanine hydroxylase's function is to convert one amino acid phenylalanine into another amino acid tyrosine. Okay, phenylalanine has to be converted to tyrosine. This function is performed by phenylalanine hydroxylase normally in normal individuals. But over here, the gene that produces this enzyme is mutated. Since this enzyme is not formed, it's not functional, this conversion does not happen. What happens when PAH is not formed? Phenylalanine, what happens is, it will get accumulated and it will get converted to another molecule called phenyl pyruvic acid and its derivatives. It will start accumulating in your body, it will start accumulating in your brain, causing mental retardation. There's so much of it present in your body that it even gets excreted through your urine, which is why it's called phenylketonuria. Okay, so phenylalanine is accumulated and converted into phenyl pyruvic acid and other derivatives. So if this gets accumulated in the brain, it causes mental retardation. Remember how I told you one of the effects of phenylketonuria is mental retardation. It can also be excreted through the urine because it's not absorbed properly by the kidneys. This also, this is an autosomal recessive trait. Previously, whatever we saw is X-linked recessive, both the color blindness and hemophilia. This one is autosomal recessive, means it will equally affect males and females. Inborn error of metabolism, it's called. Inborn means from the time a child is born, from then this disorder is there, like all genetic disorders. It is an error of metabolism because it interferes with the metabolism of amino acid phenylalanine, right? So that is why it's called inborn error of metabolism. Now let's talk about sickle cell anemia. Sickle cell anemia is an autosomal recessive trait. I'll tell you what happens in sickle cell anemia. Okay. First, let's understand how this can be transmitted. Wait. You know hemoglobin, right? In RBCs, you have a pigment called hemoglobin. You have millions of them present in each RBC. This is primarily involved in the transport of respiratory gases, oxygen, and to an extent carbon dioxide also. It's involving in transporting across your body. Now, in this hemoglobin molecule, you have 4 polypeptide chains. You have 2 alpha and 2 beta that are held together by heme group in the center. In sickle cell anemia, the beta globin chain is different, unusual, not normal. It is kind of abnormal. Why is it abnormal? The gene that is responsible for the beta globin chain production is mutated. How exactly it is mutated? You know, in any polypeptide, you will have a series of amino acids. The amino acids have to be in the same sequence for the protein to fold properly, for the polypeptide to fold properly. In normal individuals, at the 6th position, you will have glutamic acid. But because of a point mutation here, glutamic acid in the 6th position of beta globin chain will be replaced with valine. Okay. As a result, the beta globin chains are abnormal. Alpha is normal, beta is abnormal because of a point mutation. Okay. So, here for that hemoglobin molecule, you have 2 alleles, HbA, HB superscript A and HbS. HbA is the normal allele. HbS is the defective allele. Over here you can see there are 3 possibilities for genotype. Both HbA, HbA, this should actually be superscript, but there's a typing error. When both are normal, obviously the individual will be normal. When both are defective, obviously they will show symptoms of the disease. But when there is one normal and one defective, that means it is a carrier. Previously in X-linked recessive inheritance, X-linked recessive disorder, I showed you how only females can be carriers. Males, there is no possibility of being a carrier because they have only one X chromosome. In autosomal recessive disorders, there is no such thing. Males and females, both of them can be carriers because the defective gene or the gene that is responsible for this disorder or trait is present on the autosomes. Both males and females will have the same set of autosomes. So, both of them can be carriers. Okay. So, when does an individual get sickle cell anemia? Mother also should have the defective allele. Father also should have the defective allele. So, when they are carriers, it is possible for them to have a child with sickle cell anemia. Okay, so this mother is a carrier. Father also, let's assume, is a carrier. They will both produce 2 types of gametes. They will both produce 2 types of gametes, right? Because it's heterozygous. HbA, HbS, HbA and HbS. So you get HbA, HbA. This will be normal. You have HbA, HbS. This is carrier. You have HbA, HbS, carrier, HbS, HbS. They will have sickle cell anemia. So you have a 25% probability of having children with sickle cell anemia. All right. Now, I told you it is a problem with the beta globin chain. Normally, we will have glutamic acid at the 6th position, but over here, glutamic acid is replaced by valine because of a point mutation. You've learnt about it, right? How exactly the mutation happens and everything, I'll tell you in the next chapter. Now, what happens because of that one amino acid being changed? Whenever there is low oxygen conditions, either the person who has this disorder is exercising vigorously, not breathing properly, the body is not getting sufficient oxygen, or they are hiking in some high altitude area where there's not sufficient or let's say the air pressure is low, partial pressure of oxygen is low. In such cases, the hemoglobin molecules that are present inside the RBCs will polymerize. They will move to one region and start, what do you say, polymerizing just in one region of the RBC inside the cell, which will make the biconcave RBCs take up like a sickle shape. There is a reason why RBCs are biconcave, right? Because they have to squeeze in through the capillaries. It'll make them easier to squeeze in through the capillaries and deliver oxygen, pick up carbon dioxide and do that kind of things. Right now, because they are sickle shaped, it's possible that the sickle shaped RBCs can form like a blood clot within the blood vessels. Depending upon which blood vessel they formed a clot in, that part of the body, let's say that blood vessel was supposedly delivering oxygen to your spleen. If there's a clot in that vessel, then spleen will not receive sufficient oxygen, nutrients and all of that via blood, because blood cannot pass. And if it delivers blood to your heart, then your heart will be affected. If it delivers blood to your liver or your kidney, that part will be affected. So that can happen, right? So mutant hemoglobin molecules undergoes polymerization under low oxygen tension, causing change in the shape of RBC from biconcave to elongated sickle-like structure. When it is sickle-like structure, it kind of causes clots and block the way for blood, okay, because of its shape. That is the issue that happens, okay. So next is another autosomal recessive disorder. It's called thalassemia. This also is autosomal recessive disorder. This is also a blood disorder. We've learned about three blood disorders so far. Clotting is hemophilia, sickle cell anemia, and thalassemia is the third one. Hemophilia is X-linked recessive, sickle cell anemia and thalassemia, they're both autosomal recessive. Alright. So similar to how that is transmitted, this is also transmitted from carrier parents to the offspring. Okay. Yeah. So this thalassemia is caused because of mutation or deletion of some genes. In hemophilia, we saw, not in hemophilia, in sickle cell anemia, we saw how RBCs are formed, hemoglobin molecules are formed, but the hemoglobin quality is not very good. So that is a qualitative issue. The number, there is no problem. Abundant hemoglobin as much as required is formed, but their quality is poor, okay? But here that is not the case. This also is anemia only, but this is characterized by less quantity of hemoglobin being produced. Okay, quality no problem, but quantity is very less. So here there is a reduced rate in the synthesis of one of the globin chains. It could either be alpha or beta globin chain. If it is alpha chain, we call it alpha thalassemia. If it's beta chain, we call it beta thalassemia. Okay. Yeah. So, like I mentioned, if alpha chain is affected, you call it alpha thalassemia. If beta chain is affected, you call it beta thalassemia. Alpha thalassemia, why is it caused? Because there could be a mutation or deletion in the genes that are responsible for the formation of alpha polypeptide or the alpha globin chain in hemoglobin. How many genes are present? 2 genes are present. Where are they present? On chromosome number 16. What is the name of those genes? We are talking about alpha hemoglobin, HB hemoglobin, alpha A, HbA1, HbA2. Both of these genes are present on the 16th chromosome. Now, if there are 2 genes, obviously there are 4 alleles. Of them, 1 allele could be affected, 2 could be affected, 3 could be affected, 4 could be affected. Depending upon how many alleles are defective, the severity of the disease also will vary. If there are few defective alleles and there are normal alleles also, of course, there will be anemia, but not very severe. If there are more number of defective alleles, the symptoms also will be very severe. Okay, so for alpha thalassemia, remember it is chromosome number 16, HbA1, HbA2 are the 2 genes that are responsible. For beta thalassemia, not alpha, for beta thalassemia, the gene responsible, there's only one gene responsible that's present on chromosome number 11. This is beta, so HBB, since there's only one, you don't call it 1 and 2, it's just HBB gene that is present on chromosome number 11. Alright, again depending upon how many alleles are affected, the severity will vary. Thalassemia, sickle cell anemia, both of them will cause anemia. Thalassemia is a quantity problem. Sickle cell anemia is a qualitative problem. Alright. Now, that's about Mendelian disorders. Now let's talk about chromosomal disorders. Chromosomal disorders happen for a variety of reasons. Sometimes, normally we are supposed to have 46 chromosomes, 44 autosomes and 2 sex chromosomes normally. But sometimes humans can have one more or less chromosome than the normal, which can cause chromosomal disorders. That is what we're going to learn about in your syllabus, but there are many different chromosomal disorders that can happen, okay, for other reasons as well. So it is caused due to absence or excess or abnormal arrangement of one or more chromosomes. Why do you think a person can have more than normal number of chromosomes? Instead of 46, how can one person have 47? Instead of 46, how can one person have 45 chromosomes? That could be because during gamete formation, during meiosis, chromosomes have not segregated properly. In the gametes, ideally 23 chromosomes should be there, but if by chance a gamete has 24 or 22 chromosomes, then it could result in disorders like these. Okay, ploidy is the number of sets of chromosomes. Normal ploidy is euploidy. If there is any more or less than the normal set, then it is called aneuploidy. Okay, euploidy, it can be like haploid, diploid, triploid, 23 multiples. That is euploidy. Aneuploidy is deviation from that actual set. Okay. Yeah. There could be more than normal or less than normal. If there is more than normal, we call it hyperploidy. If there is less than normal, we call it hypoploidy. You know, hypo means less, hyper means more. Let's talk about hypoploidy. If there is 2 less than normal, instead of 46, if there are 44, then you call it nullisomy. If there is 1 less than normal set, then you call it monosomy. If there is 1 more than normal, you call it trisomy. If there is 2 more than normal, you call it tetrasomy. Okay, these are the terms used. Usually in humans, you see not nullisomy, you see monosomy and trisomy. In plants, you may see nullisomy and tetrasomy because our embryo only will not survive if there are so many differences in chromosomes. Okay, so today you have to learn about 1 example for monosomy and 2 examples for trisomy. Down syndrome is the first one. This is a case of autosomal trisomy. Normally, first chromosome to 22nd chromosome, it exists in pairs. But in these individuals suffering from Down syndrome, 21st chromosome, instead of having a pair of 21st chromosome, they will have 3 copies of the 21st chromosome. So they totally have 47 chromosomes. This can happen in males or in females. So they will have normal 44 autosomes. Normal sex chromosomes, XX or XY if it is girls or boys. But instead of normal 44 chromosomes, they will have normal 44 autosomes, they will have 45 autosomes. And that extra autosome is an extra copy of the 21st chromosome. Since 21st chromosome is extra, we call this trisomy 21 or trisomy of 21. So what are some of the symptoms of this disorder? Individuals are kind of mentally retarded. They will keep their mouth open and they're drooling. They will have mongoloid features like very small eyes. They will have a broad face. Basically, they will have mongoloid features. Any part of the world you take Down syndrome affected individuals, their facial features will look alike. They will have a characteristic horizontal crease on the palm of their hands that is called the palmar crease. And I told you about mental retardation. Of course, mentally they are retarded and there will be slow learning and psychomotor development is also highly delayed. They will be short. They have short, small, round head. Tongue will be furrowed. Mouth will be partially open and they're kind of drooling. Palm is broad with a characteristic palm crease. Physical, psychomotor, mental development is retarded. Why is it called Down syndrome? Does that have anything to do up-down direction? No. This was first described by a person called Langdon Down, so it's named after him. Alright. Now let's talk about the second chromosomal disorder, which is Klinefelter syndrome. This happens only in males. So normal females will have 44 autosomes plus XX. Normal males will have 44 autosomes plus XY. Klinefelter's individuals will have a total of 47 chromosomes. They will have 44 autosomes like normal people, but they will have XXY. 3 sex chromosomes will be present. They have 2 X chromosomes, so they will have some female-like features. They also have a Y chromosome, so they will have male-like features also. So this is an example for an allosomal trisomy, where there is an extra sex chromosome than normal. Totally 47 chromosomes like Down syndrome, but in Downs it was an extra 21st chromosome autosome, but here it is an extra sex chromosome. So phenotypically these will be like males. Okay, so the overall if you see, they will have masculine development, but there will be certain secondary sexual characters of females also that they show, like development of breast, which is called gynecomastia. One thing you should remember here is that these individuals are sterile, they cannot have their children, they cannot reproduce. Okay. Turner syndrome is an example for allosomal monosomy. This happens only in females. Okay. So here normal females, we have 44 autosomes plus XX2 X chromosomes. But these people with Turner's, they have 44 autosomes, but they have only one X chromosome. XO does not mean there is an O chromosome. It just means there's only one X chromosome. So they will have one chromosome less than the normal individuals. Totally there'll only be 45 chromosomes, and they will have only one sex chromosome. So such individuals are very hard to distinguish from the normal population. Characteristic difference you don't find. Compared to the other females, they will be slightly short statured. Some of them can have like a webbed neck. I don't think they are mentally retarded or anything, no such thing, but they are sterile. They cannot have children of their own. They cannot reproduce. Since they have only one X chromosome, they will not show any secondary sexual characters that females show. Okay, so that's about Turner syndrome. With that, we've come to the end of the session finally. If it helped, please let me know in the comments. If you have doubts, you can ask away in the comment section. I will answer them as soon as I can. I will see you again in the next class, the next week, with the chapter Molecular Basis of Inheritance. Until then, take care of yourselves, stay healthy, stay happy, keep studying. All the best. Thank you.